Hello, and welcome back to the Mechanochemistry discussions. This seminar series is hosted by the NSF-funded Center for Mechanical Control of Chemistry, or the CMCC. I'm Ashley Martini. The goal of our seminar series is to bring the community together. The seminar is streamed live on the third Thursday of every month at 10 a.m. Central and are available to watch anytime on the YouTube channel. I encourage you to check out the YouTube channel to watch all of the excellent presentations we've had so far as part of the mechanochemistry discussions. Also, we have some excellent speakers upcoming in the seminar series, and we hope that you will join us for all of them. Before we get started, a big thanks to Dr. James Batiste, the Center Director, Jennifer Belsick, the Center's Administrative Coordinator, and two CMCC students who enabled the seminar series, Quintarius Moore and Noah Sheehan. Thank you so much for joining us. Please do follow us on YouTube and check out all of the seminars, as well as on Twitter. Before we get started, a few quick guidelines. Note that the seminar is being recorded. However, if you have questions, you can either post them on YouTube or you can email them to us at cmccdiscussions@gmail.com, and your questions will be propagated to the speaker to be answered at the end of the presentation. Finally, it's my pleasure to welcome today's speaker. Dr. Kerstin Blunk is a professor at Johannes Kepler University in Linz, where she heads the Department of Biomolecular and Self-Organizing Matter. Her research interests combine her background in biochemistry and biophysics with the goal of developing mechanoresponsive protein building blocks for biological and material science applications. She's the co-founder and vice chair of the first Gordon Research Conference on Multiscale and Mechanochemistry and the Gordon Research Conference on Multiscale Mechanochemistry and Mechanobiology. Please join me in welcoming Dr. Kerstin Blunk.
Okay, so it seems it's time to start. I would first like to thank Ashley for this nice invitation. I'm actually really excited to share work of my group in this community. And yeah, when I went through the list of previous speakers, I realized that I'm possibly the first person here to speak about a more biology-related topic. So I will obviously talk about the molecular control of mechanoresponsive bioinspired hydrogels, but I will also give some background why we are actually interested in this, and also why we choose certain molecular building blocks actually to achieve this goal. And so let me start with some really broad background behind the work that we are doing. So we are interested in cell material interactions and primarily in the mechanical aspects that are determining cell function and actually how cells mechanically measure properties of their environment and how they respond to it. And if you all think back about biology that you had at school, maybe at some point, then everyone learned that cells and biological systems in general are exposed to a large number of biochemical signals, that there are specific interactions and signaling cascades, and that cells somehow measure this and then integrate it, and that this ultimately affects gene expression and then cell response. What has been more recently discovered is that also a large number of physical signals are actually having a similar role and also affecting cell function. And many of them also have a mechanical component. And so there are relatively simple things like the topography of the surface or material that cells grow on or grow in, and then also densities of ligands that cells can interact. But it's also elastic and viscoelastic properties of their environment, for example. And this is exactly the aspects that we are interested in more detail. And so if we think a bit more about how cells then really do that, how they can actually check what the mechanical properties of their surroundings are and which molecules are involved, I put together a slide here which gives some of the basic concepts. And so we have large number of molecules that bind other molecules, obviously, and this binding is somehow then affected by the force acting on it. And one category which has been discovered over the last 20 years are catch bonds, and that's kind of a counterintuitive thing, because if the catch bond forms and forces acting on it, actually it's being reinforced by the force acting on it, which probably for most other bonds, it will be the opposite. So bonds become weaker when you pull on them. And then there is a large number of cryptic sites, and this can be that binding sites are being exposed when a molecule is being stretched or molecule gets cleaved because it's partially unfolded, and then an enzyme can bind there and cut it in half. There are also cryptic catalytic sites, so that's kind of the opposite. Yes, when enzymatic activity is switched on because a molecule is partially unfolded and the active site is becoming exposed, and so on. And then there are mechanoresponsive, mechanosensitive ion channels, and obviously you all probably know molecular motors in our cytoskeleton, which are necessary that we can move. And also some of them actually have some feedback loops so that they can also sense forces and then somehow show some response to that. So there's a huge number of mechanisms that biology came up with to actually convert mechanical signals into biochemical signals that then also can probably be processed inside the cell. So partially inspired by this, and also because we want to understand it better, we are more interested in artificial molecular force sensors that we can actually use to interface with the cells so that we can see what they are doing. And all these examples I put here are not necessarily those that we are working with, but it's mostly the 6 maybe most well-known categories of what people are employing. And maybe in the community listening now, you may all know mechanochromic dyes, that when you pull on a small organic fluorophore, other dye, it can change its structure and you can detect a change in the optical properties. Has been discussed a lot if this also works with fluorescent proteins, and people have attempted to do this. It's not really fully established yet. There may be some candidates where this works, maybe in other cases it may be more difficult. And then again, I think for chemists, it's easy to understand that if you have a fluorophore binding to something, maybe in some cavity, and you pull out the fluorophore, then it will be released from the quenched state and you can detect some fluorescence. There are also chemical examples and counterparts to cryptic catalytic sites. So what biology does has been kind of mimicked with some very elegant chemistry over the last years. And then I think also this entropic spring principle is quite well known, right? You use a polymer which is coiled up in the relaxed state. When you stretch it, you extend it, and if there's a pair of dyes coupled to it, which does first a resonance energy transfer, you can also measure the extension of the polymer in addition with a fluorescence readout. And last but not least, and that's what I'm going to show you in the following, is you can take 2 different bonds of interest. One is molecularly characterized to know exactly its mechanical stability, and the other one is unknown. You can set up an assay actually to compare the mechanical stability of these bonds and read out the outcome of this also with a fluorescent reporter system. And so there's also lots of effort going on actually at the interface between chemistry and biology to develop these kind of mechanosensitive molecules. And some of them may work for our application and for applications that you may be more familiar with. And I think there's a lot of interesting overlap also happening between the communities. And throughout my talk, I think you may realize that even though sometimes it may be very biological, that there are similar questions and concepts that actually the biophysics community and mechanobiology communities are actually dealing with. And so if we then look a bit more at what we actually do when we want to go towards this direction of mechanoresponsive hydrogels, is that we choose a molecular force sensor. I will give you 2 examples later, what we think is suitable for a given application. And there is a few criteria, obviously, that are relevant for us. One is the force range that we either know already from the literature, or we put a lot of effort actually in quantifying that. Then we think of a readout technique, how we can see the response of the sensor. At the moment, this is entirely optical, but you could obviously also think of other readout mechanisms. And then we need to care, obviously, about the environmental conditions where the sensor should be functional, because many of these mechanochromic dyes, for example, that are known, they won't work in aqueous environment, which is obviously important for us if we think about cell culture applications. And so once we've chosen that, then we usually characterize these sensors really at the molecular level to understand the mechanism. We also quantify the forces. And if we have an optical readout, you may also think of really calibrating the optical signal versus the force, but this depends also on the mechanism of the sensor, obviously. And so for this, we use a lot of single molecule force spectroscopy. And the reason why we really study the mechanism also is that we want to have a range of tunable sensors where we make modifications in these molecules. And for this, we obviously need to know how they function so that we can make specific modifications. Then the next step is maybe a bit more obvious. We need to integrate the sensor, obviously, in our system of interest. And this is then primarily really chemistry questions also that we are dealing with. So where we have to decide what should be the density of the sensor, what do we want to read out, and which chemistry we can use. And then last but not least, obviously, we are talking about our application. And so let me start with the first example of a force sensor, as I promised. So DNA has been used as a force sensor for a long time, and there's many nice examples of this. And this is also how I got into this topic initially. So this actually started around the time of my PhD, where my supervisor was an expert in single molecule force spectroscopy. And this is obviously a very nice technique to measure forces at the molecular level, but it's also slow. Yeah, and it takes a lot of time to collect all the data. And also sometimes the measurements are noisy and you don't have the force resolution that you would like to have and so on. And he was thinking of a strategy how this can be improved and how possibly force measurements can be done in a more parallel way. But before I show you the parallel way, for those of you who don't know, let me first also explain how the single molecule force spectroscopy works in general. So here we have two pieces of DNA that are able to interact with each other. And one of these pieces is immobilized on the cantilever of the AFM, and the second piece is immobilized on the surface. Then the cantilever is lower down on the surface, and usually after a short waiting time, the DNA would bind to each other. And this is a statistical process, so sometimes you get an interaction, sometimes you don't. But after this waiting time, the cantilever, no matter if an interaction is formed, is retracted from the surface. And if there was something that has bound, then the molecule now is being stretched. The cantilever is moved away from the surface, which actually makes the cantilever to bend downwards, which can then be detected. So here we then see this increase in the force, which is acting on the cantilever. And at a certain force, then this interaction breaks and the cantilever relaxes back to its equilibrium position. So this is still a method that we use a lot also for the calibration of our molecules. But now let's say we know this, we have this blue molecule here calibrated, we know exactly how it responds to the applied force. And here are also two examples of DNA that has been characterized a lot. Yeah? We can use different pulling geometries, so we can pull on DNA in the shear geometry here, which depending on the length and the sequence of this piece of DNA, it will break somewhere between, let's say, 30 and 65 piconewton. On the other hand, we can pull it apart in a zipper-like geometry where we can break one base after the other. Then one usually gets lower forces, somewhere between 9 and 20 piconewton. So this is also something very interesting, which maybe some of you will immediately realize, but for others it's maybe not so obvious. So we have two identical sequences here now, and they have the same thermodynamic and kinetic stability in the absence of force. But by changing the attachment points, actually where we break the molecule apart, we can actually utilize it in different ways because it will rupture at different forces. And this is a very nice aspect of also using these biological molecules, because you often have the possibility to actually make use of exactly this principle. And so now coming to this parallel and more sophisticated and faster way of measuring forces in parallel. So on the top, we have our standard AFM setup. So here now we are interested in the properties of this red molecule here, and our force sensor is actually the bluely stained AFM cantilever here. And obviously we can now characterize this, measure this repeatedly to get some statistics. But on the other hand, we could also say, okay, we're interested in the mechanical properties of this red molecule, and we have a blue one, which we already calibrated before, and we know exactly at what force it breaks. And so we can then put together this chain of molecules and stretch the two molecules in series. And then the weaker one will break with a higher probability. And then the only thing you need is actually a readout to somehow measure this probability, which one of these bonds breaks more easily. And this can be done by attaching a fluorophore also in the center of this chain of molecules, and then simply by doing many of these tests in parallel, so that we can then really compare what is happening here by measuring how many fluorophores end up on this top surface and how many fluorophores end up on the bottom surface. And this will directly reflect the relative stability of the two bonds that we are pulling in series. And here is a bit of a better idea of how this works practically. So we immobilize all this chain of molecules on a glass surface. And again, you see this here at the bottom. And then we have a molecule which can actually hook up to this chain, which is immobilized on a PDMS stamp, which has a micro pattern. And so this is actually an experiment which is very similar to micro contact printing, except that it's done in liquid. And so then if you have this contact here established and you pull off the PDMS, then all the molecules that are immobilized are mechanically loaded. And then we just need to check how much fluorescence ends up on the top part of the PDMS part and how many molecules remain on the bottom part, basically. And here are two examples. So here exactly we made use of this pulling geometry effect. So here now the blue one and the red one is exactly the same sequence, just that the pulling geometry has been changed. So in the left side, we compare DNA in this unzipped geometry with one in the shear geometry. And on the right side, we compare 2 times the unzipped geometry. So if we start with that, we would now expect a 50-50 distribution of the fluorescent molecule on the top and on the bottom surface, because we have a symmetric system. So the probability of rupture for both the red and the blue should actually be the same. And that should now change on the left side, because now the DNA in the shear geometry is much stronger than the DNA in the unzipped geometry. So if we measure fluorescence on the top surface, we should have less fluorescence here than we have for the symmetric setup here. And that's exactly also what we see when we measure the fluorescence intensity on the top surface and calculate some ratios. Then we see that we get at least 50 times more fluorophores on the top surface for this geometry here. And then we wanted to know also what is actually the sensitivity of this, because here the differences are quite obvious. I showed you the forces before. And here now we wanted really to find out what the absolute limit was. So we made one piece of DNA which has one base was actually mutated. And this is something I have to say, which people tried to distinguish with AFM, the red and the blue one, and they failed to do so. But if we do this direct comparison, now we actually do see a difference between these DNA molecules. It's only a factor 1.7, but still we can really see that there is some difference in mechanical stability. And so the reason why this actually works is that we are now directly comparing two things. So the same noise applies to both systems, basically. And so we're actually using kind of a balance where we just see, okay, which one, let's say, is more heavy or which one is stronger. But when you use an AFM cantilever, it's more similar to this setup on the left, which is a scale where you actually have to measure first one of them and then the second one independently. And if there is noise in the measurement, then you may not be able to distinguish this, because the difference may be hidden actually in all the noise of this measurement and the measurement error, obviously. So this is a clear advantage of doing this direct comparison between two molecules. And yeah, so the nice thing for me was as I said, this was a project that started during my PhD and then went on a little bit afterwards. And then at some point, really other people picked up this idea and started to use this, for example, for cell culture applications. And the goal of this project here was, which was done in the group of TJ Ha at Johns Hopkins, they now also use short DNA molecules the same way as we did here also in this unzipped geometry and also in the shear geometry. And they also came up with a few intermediate geometries so that they could actually establish a range of sensors which all have a different mechanical stability. And so the other important thing for you to know here is that, see, this is the one side where it's immobilized to the surface, and here this top part, these green little spheres, these are short peptide ligands that cell surface receptors can recognize. So they would actually bind here to these little green spheres. And cells then, when they bind with these receptors, they start to apply forces to their environment. So they generate forces in the cytoskeleton, which is then transmitted to the outside, to the surface where they are growing. And they actually do this to find out what the mechanical properties of the surface are. And what you can then see here on the bottom is that if we have these weak sensors, the cells actually have difficulty to attach to the surface, and they are only able to grow on these surfaces if this sensor here can survive at least 43 piconewton, according to their calibration. And there seems to be a really clear cutoff also. So what this now tells us is that when we have this, let's say, this nice chain of one-to-one stoichiometry from the surface to the cell surface receptor, that there's at least a force of 43 piconewton acting on the receptor. And this is now a tool that people are using more and more to understand, for example, if mutated cells, for example, cancer cells, have a different mechanical profile and if they apply different forces to their environment, which could possibly on the long term become some kind of diagnostic tool. And so the group of Andreas Walter, they then also used DNAs to make hydrogels. Actually, these hydrogels are purely based on DNA molecules, and you see the concept down here. So they have a few long chains which form complementary strands which act as crosslinks, and then they have this pink sensor here in the center. And also here, this is again a pulling geometry thing, because all these other DNA interactions here, if you look at it, they're all loaded in the shear geometry, but the purple one here, when the hydrogel is being stretched, is loaded in the unzipped geometry. So this is kind of the predetermined breaking point in this material. And what they did in addition is that they attached a fluorophore and a quencher. So when this is zipped apart, then the quencher will be far away from the fluorophore so that this can be detected. And they also put a reference fluorophore that they can really compare the changes and normalize it to the hydrogel concentration or thickness or whatever is needed. And yeah, this is basically what would happen then if this material is being stretched. So this is all very nice and made me very happy when I saw it, but now obviously you're already suspecting that I also have some criticism here. And so one thing that I would criticize is that if you want to use this for cell culture applications, then you may question if DNA is the right molecule for that, because obviously it's an intracellular molecule which is meant to store the genetic information and is involved in protein expression and so on. But usually it only gets to the extracellular side if something's wrong, when cells are dying or something related. So this is maybe not what you would want to use for cell culture experiments. And also this 2D cell culture experiment I showed you on the previous slide is also very important and has probably lots of applications, but most cells live in a 3D environment and they also behave differently if you grow them really in a 3D hydrogel compared to a 2-dimensional glass surface, let's say. And so would obviously be nice to somehow address these two limitations and come up with a maybe even more flexible force sensor system for developing this further. And this is what we have been doing over the last years. So we replaced the DNA force sensor with a protein-based force sensor here, which consists of alpha helices. I'll explain this to you, what that is exactly on the following slides. And again, here we can introduce a fluorescent reporter system where we have high acceptor fluorescence when the sensor is intact, and where we have low acceptor fluorescence when it's broken apart. We can then use these directly as crosslinks in hydrogels, and ideally, obviously, keep the fluorescent reporter system there so that we can see the status of the hydrogel. And then obviously we can hopefully at some point in the future grow cells in this to then directly visualize how the cells interact with these materials. And just to give you a bit of an idea also of the techniques that we are using. So here as I said, these are protein or peptide building blocks. So we usually design them from scratch with some inspiration from the literature, also to introduce modifications so we can tune the force range. Then we do, for calibration, single molecule for spectroscopy, and have a few collaborations also with people who are doing molecular and dynamic simulations to really understand what happens when a force is acting here. Then we spend some time on synthesizing different types of materials to also understand how they can be tuned. And our main characterization method right now is rheology, but obviously we have to combine this with fluorescence readout in the near future to actually see really the force response. And for now, we are doing some 2D cell culture, and the rest is to come once we have full control over our hydrogels here. So what are these alpha helical protein building blocks now that we are using? So these guys here are called coiled coils. I think we can easily imagine this when you look at their structure. So we start with some alpha helices which interact with each other, and the whole assembly forms this super helical structure, which you see best here in this front view at the bottom. And so why did we choose these actually? And the main reason is that they are frequent building blocks in the cytoskeleton and the extracellular matrix, and also a large number of other biological materials that often also have mechanical function. And what you see here on the left is actually a list of extracellular matrix and cytoskeleton proteins, which all have these coiled coils as building blocks. And for us, this is a good starting point, obviously, because we are using something now for our materials and for sensors that the cells also experience in their natural environment. And I also have to say that when we chose them, there was not much known in the first place about their mechanical properties, even though they have such an important mechanical function. So we were also, from this point of view, very interested in characterizing these in more detail. To give you a bit of a more detailed idea of how these structures actually form, we need to look at the amino acid sequence. And most of these coiled coils, they consist of repetitive blocks of 7 amino acids. They're also called heptads. And they're always specific amino acids with specific properties in these A and D positions, for example. And here they are hydrophobic, and when the helix folds, they actually all align on the same face of the helix to form this hydrophobic core here, which is the main driving force for the structure formation. In addition to these hydrophobic amino acids, there are some charged ones also, which are actually lining this hydrophobic core and can provide additional stability. In our case, we mostly use these charged amino acids to ensure that we have a heterodimeric system. So we actually have a positively charged and a negatively charged helix, which allows us to handle these peptides that we are using individually, so that only the coiled coil structure forms when they're both mixed together. This gives us much more control over our experiments. And then, as I said, when we started this, there was nothing known really about the mechanical properties of these coiled coils in the shear geometry. For the unzipped geometry, some AFM data was already published. But obviously, if you want to play with these pulling geometries, we also need to understand what happens in the shear geometry. So we took the sequence that I just showed you and extended it with a few amino acids here, like especially these cysteines at the termini that we can use for site-specific immobilization. so that really the pulling geometry is also defined. And then we took one of these peptides, immobilized it to the surface, and the second one to the AFM cantilever, very similar to what I showed you for the DNA before. And here at the bottom, now you see a typical force extension curve that we get from this experiment. What I need to mention is that the majority of this force extension curve here, this part, actually comes from these polymeric spacers, this polyethylene glycol that we are employing to get some linker between our coiled coil and the surfaces. And it's first of all there to improve the specificity of the binding, but second, also we can use this signature here for the polymer stretching, also to identify if we're really pulling on the right system, because these PEG spacers have a relatively defined length, so we can actually use the length until the rupture occurs as an indication if we are really pulling on the full chain. And then, yeah, so here this is the rupture point, obviously, where the coiled coil dissociates apart. And these forces is what we are mostly interested in. So since this is a statistical process, we are not done, obviously, with measuring one force extension curve. So we usually collect hundreds to thousands, and then select them to really identify those here which rupture at the expected length. And then we can construct these histograms. And this is one example histogram, which was collected at a relatively low cantilever retract velocity. But we then also repeat this at 6 different velocities. And the reason why we do that is that it's well established in the biophysics community, but I think also mechanochemistry community, that if you pull faster on a receptor ligand interaction, you need more force. And this is also what you see here. So with increasing loading rate, then also the forces actually shift to higher values. And these values we can then also use for further analysis of our interaction. And here is just 2 examples. So the black curve is for the sequences that I showed you. This is 3 repeats of the same system. And the blue one is another coiled coil where we cut off the piece at one terminus to destabilize it a little bit. And so this helped us then to understand more also what happens mechanistically. This is the equation, this Bell-Evans equation that is usually used for analyzing these data. This is really well established for a long time. And what you get from this is the thermal dissociation rate if there is no force acting on the complex. So this is extrapolated basically to force-free conditions. And in addition, we get this delta X value, which is the width of the binding potential. I will show you more detail on the following slide, actually. And so this is what we get for these 2 systems after we have fitted the data. So first of all, we obviously have our bound state here where the coiled coil is intact. Then we pull it apart. It has to get across this energy barrier here to get to the unbound state. And then there is some transition state in between where possibly the helix is partially unfolded. And then we see that for the truncated version of the coiled coil, we have a faster dissociation rate, which possibly makes sense because there's less interaction between the 2 chains. And what we also see is that the width of this potential here, so the distance from the ground state to the transition state, is also shorter for this truncated sequence. And the easiest way to interpret this delta X, in my opinion, is that you imagine how much the molecule can be stretched before it's destabilized such that it can dissociate. And maybe here you can also imagine that if it's a molecule which has, let's say, less helical turns because we truncated it, then it tolerates less deformation before it's destabilized and falls apart. And this is what is also reflected in this delta X value here. And so we obviously wanted to understand a bit more what is going on and also how the transition state possibly looks like, because as I said, we want to use this as a molecular building block that we can also tune, so where we can make specific modifications and then change this range of rupture forces that we measured. And we thought that the best way of doing this is to perform molecular dynamic simulations, where we also pull on these molecules to actually get an idea of what is really happening to the structure. And to our surprise, we could not make sense of what we saw in the simulations and in the force spectroscopy for a long time, because somehow the mechanisms seem to be completely different. What we saw in the simulations at high velocities was that the molecules would start unraveling from the ends where the force is applied. And if we then extrapolated from the different loading rates that we did in the simulations to what happened in the experiment, there was just no overlap between the data. And then we did simulations also at slightly lower velocities and tried to go as low as we can possibly go with the simulations. And then we actually saw a change in the mechanism also. And this change was interesting in that sense that at the time scale where it's known that alpha helices are refolding, we actually didn't see this continuous unraveling from the ends anymore, but a relative displacement of the two helices with respect to each other, which suggests that they maybe locally unfold and then move a little bit and then refold because it's a repetitive sequence kind of, and that also this intermediate state can be kind of metastable, and that this is something we see in the simulations. On the other hand, in the experiment, this metastable state is probably not stable enough that we can really probe it in the force spectroscopy experiments. And we think that somewhere this structure then dissociates, and that this is what happens in the AFM experiments, basically. So on the one hand, this was nice because we saw that probably the helix stability is a key parameter, because if we could somehow stabilize the helices, we would probably need more force to initiate the unfolding. But on the other hand, it was not really possible from this to predict which position in the coil you may have to modify to really change the response mechanism. And so we tried a few things. We tried a lot more things. I just want to show you some examples here. So we talked about the length already. So this is clearly a parameter, but maybe it's also something obvious. So the more interesting aspect is that we played with the stability of the helix. So we decreased the helix propensity of the chain by replacing alanine with serine in every heptad. And this actually really led to a lower mechanical stability, which also proves this hypothesis we had after we did the simulations, that the helix stability is really an important parameter here. We also managed to stabilize the helix by putting some histidines in the sequence, which could then bind nickel and actually bridge helical turns. We put this in different positions, directly at the force application point, at opposing positions, and so on. And then in any case, we always saw a small increase in the stability of the structure, which clearly again shows that the helix stability is a key factor, but unfortunately it also didn't give us so much of an increase as what we were hoping for. Another parameter that can be played with, obviously, is the hydrophobic core. Also, if you replace a very bulky amino acid with a less bulky one, then we also saw big differences in the rupture forces, but then also the width of the potential and other parameters did actually change. So this is the library we have right now, plus lots of other sequences that I'm not showing you because it may also get too boring. What we wanted to take in the next step with the library available was that we wanted just to repeat the cell culture experiments that I mentioned before with the DNA to actually see if this strategy works, if our coils are actually stable enough to maintain cell attachment and so on. And this is just a very preliminary result where we can clearly see that after 30 minutes, the cells are able to attach on the surface, and that also after 120 minutes for this one sensor that we tried here, they don't seem to be very happy anymore which is actually not the case for the control cells. Yeah? So possibly with the sensors we currently have, we are also just at this threshold force that we saw in the previous experiment. So this is work that is currently ongoing. We are now also starting to play with different pulling geometries here, so that we can, and also with fluorescently labeled cells, so that we can really see in more detail what is going on here. Obviously, we want to take this to a 3D cell culture experiment, as I mentioned before. And here we first asked ourselves the question, okay, so now let's say we use these 4 sensors in a hydrogel. What does actually happen to the hydrogel? So will the 4 sensors really be activated? Will they somehow affect the material properties? And what are we actually doing to the cells when we're really able to move to this step? And the best experiment we came up with to check for this was to use a special type of hydrogels, which is based on star-shaped polyethylene glycol. So this is basically one star here. And then we attach our coil-forming peptides to the ends of these stars. And the key message here is actually that when we look at this little blue box here, this is now one network chain, basically. And we have the core of the star pack here, and we have the core of the second star pack there. And then we have one chain, which is then cross-linked with this coil. And what we did is that we chose exactly the same length for each one of these polymers here, these arms on the star pack, as what we had as a spacer in the single molecule force spectroscopy experiment before. Which bottom line means that what we did in the force spectroscopy experiment was to pull on exactly one network chain. And like this, we thought we can then really compare the results without being too much affected by effects from the polymer, which would then in turn affect the strain rate and so on. And obviously, then we compared our single molecule force spectroscopy data, or that's what we are currently doing with what we get from rheology data, where we try to break the material. But let me start slowly until we get to this point. So what we do is that we take star packs, which have a maleimide functional group, because our peptides have cysteines, if you remember. And then we functionalize all the arms so that we get 2 different types of stars, one with peptide A and one with peptide B, and then to some extent we just have to mix them in a 1-to-1 ratio to get our hydrogels. In the first step, we just did linear rheology to know about the linear viscoelastic range and also how actually these dynamic crosslinks now affect the relaxation properties of the material. So this is a typical amplitude sweep. I'm not sure how much you're familiar with rheology. So what basically happens is that you take the hydrogel and put it between 2 plates. The bottom plate is fixed and the top plate can oscillate. And it can oscillate with different frequencies and with different amplitudes. And what you do when you do an amplitude sweep is that you keep the frequency constant, in this case here at 1.6 per second, and the strain amplitude is varied over 3 orders of magnitude. And what you then see here is that the material seems to have the same properties up to an amplitude of around 100%. And then after that, something happens. And so I need to explain maybe also a bit more what the storage and the loss modulus is. So the storage modulus reflects the elastic component of this viscoelastic material. So in the frequency range from here to around 100%, it acts like a solid, which means that we have an intact hydrogel. And the loss modulus reflects the viscous or liquid component of the hydrogel. And then if we approach here around 300% strain, then the loss modulus is higher than the storage modulus, or here then we are dealing with a liquid material, or at least the liquid properties are more dominant, which suggests to us that we now have broken something in the hydrogel. So something is not the way it should anymore. So what this tells us now is that we have actually a relatively large linear viscoelastic range. And we also tried this with different length of our pack arms. And then you see actually that this crossover point is also shifted depending on the length of the pack arm. So this is a first indication that in this experiment, we actually really break the crosslinks when we do this. So that whenever the polymer arms are extended to a certain length, then the coil will actually break. So the second thing that was interesting for us, and I will talk about this in a bit more detail, is what happens when we test the relaxation time of the material. And this is not the opposite experiment, so we keep the strain amplitude constant, and it is actually in the linear viscoelastic range where we don't destroy the hydrogel, and then we vary the frequency of the oscillation. And what you see here now is that we have a storage modulus, which is kind of constant, also across a relatively large range of frequencies. And at low frequencies here, it shows a crossover at some point. And below this crossover, the loss modulus is dominant. So we are kind of at low frequencies, the rheometer thinks the material is a liquid, basically. And this is also directly related to these crosslink properties, because if you manipulate the material in a fast time scale, the crosslinks will be intact on this time scale of the manipulation. So it feels like a solid. If you manipulate the material on a time scale slower than the crosslink association and dissociation rate, then the material will feel like a liquid. So this crossover point here is somehow related to the kinetics of the crosslinks. And for some time, well, I think it's still not fully clear how exactly it relates, if it relates to the on rate or the off rate or the combination of the two. And this is something that we then also investigated in a bit more detail. So we wanted to tune the relaxation time, basically. And we wanted to tune this without doing too many changes to the network itself, and also ideally maintaining the same crosslink with, say, relatively similar thermodynamic and kinetic stability. So what we did is to make use of this idea that the helices start to unfold at the points of force application. And to prevent this, we introduced these histidines, as I mentioned before, because what this now allows us to do is to form a hydrogel based on the coil-coil crosslink, and then actually manipulate its stability by adding nickel ions, because when the nickel ions bind here, this terminus gets stabilized. And when there's no nickel bound, then we have a slightly less stable system. And this is then what you immediately see in the relaxation time of these hydrogels. So this is without nickel here, the blue curve, we have a relaxation time around 100 per second. And if nickel is added, the system gets stabilized and it shifts. So again, this shows that we have pretty good control over this material, and that the crosslinks are the main units which are responding to the changes that we make to it. And then we wanted to go even one step further to understand really what happens there at the molecular level. So we replaced this crosslink here, where the nickel actually bridges in the same peptide chain, by this sequence where we now have histidines in a different spacing. So now this intra-chain cross-linking here can actually not happen anymore because the distance is too big. So now it can only interact with neighboring molecules to form these kind of structures here. And in this case, we used zinc. And what you then see is that we have, here's our relaxation time without zinc. Here we have a roughly 1:20 ratio of histidine to zinc. So you already see a pretty big shift in the relaxation time. And if we have a 1:1 ratio, there is no crossover at all anymore. So we have more or less elastic material, which doesn't show any relaxation anymore on the time scale of our experiment. And so this is now interesting in that sense that we have actually the same molecular building blocks. It's just that the network architecture has changed, because we have added these zinc ions here, which induce this aggregation. And I was kind of surprised by this. I'm not sure if you are also surprised, because as I said, many people relate this relaxation time to the association and dissociation rate of the crosslinks, but we didn't change them. Yeah, we just changed the aggregation state, which means we changed the network architecture, that now there are more chains actually connected to one network point. And this was actually enough to change the relaxation time by more than 2 orders of magnitude. So it's not so easy in that sense. So I would say possibly if you have the same network architecture, you really have a direct correlation between crosslink properties and relaxation time. But in that moment where the network architecture gets modified, then this is the dominating factor actually, and you get much bigger changes than simply what the crosslink does. And we've shown this in the meantime in a few other examples as well, which I don't want to go into more detail because there is definitely no time to do that. Because in the last maybe 5 minutes, I want to go back to this original goal just very quickly. If we can now really correlate these molecular properties with the non-linear viscoelastic properties of the material when we actually really break the crosslinks. And so to do this, we then thought, okay, so to really prove there's a mechanical effect, we need to make use of these pulling geometries again, because then we have crosslinks which have the same thermodynamic and kinetic properties, but we know they break at different forces. And like this, we can then possibly really find out if there is really bond rupture occurring in the hydrogel and if we can measure this. So this is first the control where we did a frequency sweep again, and indeed for these 2 different crosslinks with different pulling geometries, we have very similar relaxation times. And then if we do this nonlinear rheology experiment where we just rotate the top plate in one direction to very large strains until the material breaks, we really see rupture here. And the very interesting thing for us is that at high strain rates, we actually see that this rupture peak here shifts to higher strains and also to higher stresses. So we have a very clear strain rate dependence of the material response. And this is obviously now very similar to what we have in the AFM with this Bell-Evans model. And we are currently analyzing this data in a very similar way and comparing the different pulling geometries, and preliminary results show that we can actually really correlate the molecular and the material properties. But this is really ongoing work. And I also have to admit there is still a lot of things that have to be tested because, as I said, it also depends on the network topology, which is obviously another important factor that has to be considered here. And for this, we are still doing simulations and control experiments to actually see how this also affects the processes that are happening here when the material fails. And so with this, I'm at the end actually of what I wanted to tell you today. So just coming back to this first slide again. So, and also coming back to these physical signals. So I mentioned already that cells somehow manage to find out how the elastic and the viscoelastic properties of their surroundings are. And lots of experiments have been done to test that. Things that are not known is how the cells exactly do that, because the cells don't have a rheometer. They can grow inside the material and have mechanisms to test that. And the other question is also, so these are usually linear viscoelastic parameters. So the next question for us is, do the cells actually also really break bonds, and are they responding to nonlinear viscoelastic properties in the material? As we are now trying to probe also with this rheology experiment that I just showed you on the previous slide. So this is really what we would like to find out next with these coiled crosslink materials, and then also with the fluorescent reporter system that I mentioned a few times. That's something we're currently working on. It's still a bit challenging to find the best labeling position without affecting the properties of the structure, but we're definitely making progress. And if maybe at some point in the future we manage to meet on a conference, I hope I can give you some answers on these remaining questions as well. And with this, I'm at the end. I want to thank a large number of people who contributed to the work that I have been showing. Most of the hydrogel work was done by Alberto and Isabel. Actually, Melis does most of the cell culture experiment, or did most of the cell culture experiments in the group. And then also want to thank Anna, who helps us a lot with molecular dynamic simulations. Yeah, and with this, I'm at the end, looking forward to your questions, and just want to use this as a chance to tell you about this conference that I'm organizing together with a colleague, if you're not already aware of it. Thank you very much.
All right, thank you. We have a series of questions. I'll cherry pick a few of them. First of all, when you pull on a DNA or a coil, especially one subject to shear, how do you know that it's breaking at the point you identified as opposed to separating from the surface?
So this is related to the chemistry that we are using. So all the other bonds in our system, so the attachment to the surface and then the attachment of the polymer to the DNA, also this is all covalent bonds. So this is what we really make sure. So whenever we want to break something that we are interested in, we make sure that really the other bonds in the system, at least in terms of forces, are at least 10 times as strong. And you cannot fully exclude that with a low probability one of these other bonds breaks as well. But I think if the rupture forces are known to be a factor 10 different, the chances are very low.
All right, thank you. Have you explored any environmental effects? For example, temperature seems like it would play an important role given the thermal activation of these reactions.
Yes, definitely. Yeah, so temperature is important to control for us. So usually we do all these experiments at room temperature, so somewhere between 20 and 25 degrees. And we usually also characterize the thermodynamic stability of all these coiled coils before. So we measure melting temperature with CD spectroscopy so that we also know if we are close to this, if the melting temperature is close to the measurement temperature, which usually is not the case. So most of the structures we use at least have a melting temperature of 60 degrees. But it's something really important, obviously, if you do the calibration of the force sensors at 25 degrees, and let's say the cell culture experiments at 37 degrees, you need to be at least aware that this may not exactly reflect the conditions. But in principle, this Bell-Evans equation also allows you to recalculate for the different temperature, because the temperature is a parameter in the equation. So yes, it is very important.
All right. There's a related question here about the delta X term that was found to be approximately 1 nanometer. Do you have any physical insight into the meaning of this? For example, is it related to length of the molecule?
Yeah, this is an ongoing discussion in the community. Yeah, so for most proteins that are being pulled, I would say no, because obviously proteins have a very complex energy landscape and you're projecting it to this very simple two-state model, but you don't really know how it really reacts on the reaction coordinates. So for most proteins, I think people would say no. In our case, it may be a bit different, even though I would still say you should be careful, because in our case, the helices are aligned directly in the pulling direction, obviously. Yeah, so you could think that the reaction coordinate is less complex, so that it maybe really relates to the unfolding of the helices. This is one thing we try to make sense of from the simulations, and possibly it is kind of a bit related because the whole coiled coil is around 4 nanometers long. Yeah, so you are very tempted to actually say there is some real physical meaning to this, but I wouldn't want to go that far. We don't have enough evidence for that.
All right, let's do one more. This is about the hydrogel work. You showed a very distinct yield or failure. Can you talk more about the failure mechanism? For example, do chains break? Is there interchain slips? Do all these happen at once? Do you have any ideas about this?
Yeah, this is exactly what we are currently busy with, and we try to find this out in different ways. So what we currently think is that our hydrogels are not very homogeneous because we are just at the critical overlap concentration. And there are some simulations that people did which show that in this range, you probably have some clusters where the density of chains is higher, which are then maybe connected by relatively extended chains to make a network kind of. And that would maybe also explain what we see, because maybe it's really enough to break a few of these relatively extended chains to actually do major damage to the material. And we are relatively sure that it's not polymer chains that are breaking, but really the crosslinks, because when we take a different crosslink, this behavior immediately changes. Yeah? So we can really relate this to the molecular properties we measured before. We also for a long time thought we don't have entanglements because similar hydrogels that have been made with different crosslinks didn't show that, but we did some solid state NMR together with a collaborator, and they also see entanglements in these networks. So it's definitely much more complex than just a simple answer to this, I'm afraid.
All right, thank you very much. And thank you again for that presentation.
Thank you again for the invitation.
Thank you again, Dr. Blanc, for that outstanding presentation. And thank you for joining us. Again, if you've missed any of the speakers as part of mechanochemistry discussions, we encourage you to check them out on the YouTube channel. And an early heads up of a few upcoming speakers as part of the mechanochemistry discussions. We hope that you will join us for all of them. Thank you again.